very much. For me, it's my first GRIN to be, or first GRIN meeting, and I have to say I'm excited, especially because of course, the amazing science and working together with the clinicians, but also because of the families, and that's really inspiring, and thank you for inviting and for hosting such a meeting. I think that's the way science should be done. I want to begin by lowering expectations. So there was very sophisticated science that was shown here over the past 3 days, and I'm only an Uber driver. We use nanotechnology inside the body in order to take medicines to the site they're needed in. And I'll try to convince you that we can also take nanoparticles to the brain. And if you have an interesting payload, that's where we come in. We're less on the design of the payload, more on design of the delivery system. So this is my group, and I really also want to thank the To Be Cured that have been supporting us and actually introduced us to this disease, and which we were not aware of beforehand. So thank you so much, and thank you for bringing us into this community. So I'd like to start just to lay the ground, because nanotechnology was not mentioned in this meeting. So just to show where nanotechnology is today in the clinic, and to try and just set where nanotechnology actually met me. And I was an undergraduate student, and I was inspired to see this paper that was published by 3 clinicians. They were looking at different cancer patients, and these cancer patients actually had tumors in different sites inside the body, and they were injected nanoparticles intravenously, so a systemic administration, and the nanoparticles actually targeted the different tumors inside the body. So this is now an imaging modality, and you can see the 3 patients, one a tumor at the base of the tongue, a patient with breast cancer, and a patient with lung cancer. And the nanoparticles actually, after the systemic administration with a contrast agent, reach that specific site inside the body. And the nanoparticles that were used here are liposomes. These are lipid vesicles that are 100 nanometers in diameter. And today we already have next generation of these systems. Usually the medicine is inside that particle, and a targeting moiety will be on the outer surface of these systems. That's really where the engineering goes, in order to make sure that it reaches the target site and also releases the medicine where it's most needed inside the body. And the first generation actually of nanotechnologies took advantages only of differences in the permeability of blood vessels inside the body. So having, for example, a tumor or sites of inflammation, or in certain cases also the blood-brain barrier, it may be permeated. And if we take a tumor, that's the best example. Actually, the blood vessels are not continuous, and nanoparticles can extravasate out of the blood vessel and accumulate inside the tumor site. So based on size alone, having that nano dimension alone allowed the targeting of the first generation of nanotechnologies inside the clinic. And that was enough in order to develop a first set of about 80 drugs that are nano drugs that are used in the clinic. The first one was actually invented in the Hebrew University in Jerusalem. It's called Doxil. It's for treating breast cancer patients. Today it's already used very, very widely in the clinic. And the advantage actually this system had was loading a chemotherapy inside a lipid nanoparticle, circulated in the blood, avoiding the tissues such as the heart that could be damaged by the chemotherapy, and bringing that chemotherapy directly to where it's needed inside the body. So for patients, it was a great advantage. But since then, really many different ones, nanotechnologies have been introduced into the clinic, and they're administered in many different ways, such as through inhalation. But I will focus today primarily on intravenous administration. You can also, a nice thing with nanotechnology is that you can combine different medications inside the system. If you take that tennis ball, which that's the way I always try to think about it, you can actually load it with different medications, and by that get a better therapeutic activity inside the body. But if we look at it historically, around the end of 2019 was one of the first big breakthroughs in the field, where liposomes merged with lipid nanoparticles. Instead of a payload of a small molecule, now genes were actually loaded. ASOs were mentioned yesterday, but here siRNA, which is an analog, it's a short interference RNA molecule, were loaded into these lipid nanoparticles. And they went to treat an orphan disease, a rare illness in patients that have a protein that actually misfolds inside the liver. And the disease is called HATTR, and there was no treatment in the clinic at the time. 50,000 patients, so I think also number-wise, we're talking not very far than what we have with the GRIN2B. And the FDA goes for the clinical trial, and it actually stops it after 18 months. And everyone was very worried. Why did they stop the clinical trial? And then they said it's immoral to continue a clinical trial when you have such significance, such clinical significance in the clinic. What they had is these alligator claws, that's what they called it. The patients that were responding are the one on the lower line, and the patients that were not responding, were giving just the placebo, were on the higher line, and actually deteriorating their condition. So the nanoparticles actually were delivering a medicine inside into the liver, more specifically here, delivering siRNA into the hepatocytes of the liver, down-regulating or stopping the synthesis of a protein inside the liver and improving actually the outcome of the patients. And then, of course, all the patients were given the full treatment, and that was actually very, very exciting for everyone that was working in the field. Of course, it took a couple months later, so we're talking around 2020, and a COVID outbreak, and very similar particles are then used not to deliver siRNA, but to deliver messenger RNA to encode for the COVID vaccines that many of us had injected to us. And really, today, billions and billions of people have been injected with these lipid nanoparticles. And the nice thing about these systems is that they're very modular, that you can actually load different genes into them in order to address different diseases inside the body. And this is a bit what, this is just one image, a cryo-TM image of one of the COVID vaccines that was out there. And you can see the particles are around 100 nanometers in diameter. They contain the gene inside them, and these are injected and actually encode for a protein inside the body. Just another image of these systems to get a feel. And again, we're looking at the nanoscale. And the nanoscale here remains usually around 100 nanometers. That has been found to be taken up very well by cells, traffic very well in the blood, and also be able to cross the blood-brain barrier if you add a targeting moiety onto the surface. I won't talk a lot about the chemistry, but I am a chemical engineer, so I have to. And I'll just say this, that we're seeing an evolution also on the molecular side. The building blocks of these nanoparticles has actually evolved over the years. In the beginning, we used only simple phospholipids, and today we're using more and more sophisticated phospholipids in order to deliver our payloads. If we deliver a gene, we need to adjust some of our lipids so we have a high loading capacity, but also that we release them inside the site of the body or inside the cells they're needed in. So what is it about? It's really about formulation. Is this an example of a clinical trial that we've been involved in? This is a treatment for osteoarthritis, where we looked only at the lipids as a treatment modality. So again, the delivery system here without any medicine. We're in phase 3 trials now, so we've treated already more than 1,000 patients. And what we have is looking at the specific lipids injected into the knees of these patients actually improves the lubrication and reduces pain, and improves the friction or reduces the friction inside the knees of these patients. The reason I'm showing you this slide is that the engineering, and that's the site where we actually invest most of our time, is very important. One nanoparticle has to be aimed for a specific use, for a specific disease, and also for the payload. And that's where the engineering actually goes. So if we want to target the brain, the particle will be different than one that will target the liver or that will be injected intra-articularly to treat osteoarthritis. And these are just some of the results from the clinical results, which were very promising. This is in the phase 1. The phase 2 is also that, and the phase 3 is actually ongoing. So I'd like to jump forward and take these particles and now actually look not at the lipid composition of them, but actually can we reach the brain with such lipid particles? And here we actually understood that having a particle that only circulates in the blood will not be enough in order to cross into the brain. So what we did is we changed the molecular surface of these particles, and we actually connected to the outer surface of our nanoparticles a molecule that is recognized by the blood-brain barrier in order to cross the blood-brain barrier and penetrate into the brain. The molecule we chose is called transferrin because it's used very widely, and the disease model we actually chose to treat is Parkinson's disease for 2 reasons. 1, we didn't know about GRIN, and 2, actually the models of Parkinson's disease are very good for engineers. You can see a very clear outcome, delivery, activity, modality, things that I think here when I come, now I'm much more convinced that, we can also show the same also in GRIN. And actually that's very exciting. But what I would want to try and convince is that these particles can cross in. The payload here is a protein payload. I'll show later also genetic payload. But a bit just about Parkinson's disease. In Parkinson, again, there's a protein that misfolds inside the neurons in the brain. The protein is called alpha-synuclein. It aggregates inside the neurons and actually damages them. If we can actually stop that aggregation inside the neurons, we can improve the outcome of the disease. However, drugs, specifically monoclonal antibodies that were introduced to the clinic, did not penetrate the brain efficiently. So what we asked is, can we take a drug that showed to be efficient in the dish and then actually load it into our nanoparticles, our brain-targeted nanoparticles, and deliver it into the brain? So what we did is very simple. We took a monoclonal antibody which failed in clinical trials, but worked very well in the animal models. And then what we did is in the dish. And then what we did is we loaded this monoclonal antibody into our nanoparticles and tested its ability to cross into the brain. And this is what you see in the schematic. And the receptor we chose, again, is transferrin, because the transferrin receptor is actually overexpressed on the blood-brain barrier during Parkinson's disease. I think if we come to target GRIN2B, we need to ask ourselves which receptors are actually being expressed on the blood-brain barrier in order to make sure that we cross in. In Parkinson, it was very, very clear. The areas that are diseased overexpress this specific receptor, and that's why we conjugate it to the surface of our particles. And the particle looks like this. It's about 100 nanometers in diameter. It has the transferrin on its outer surface, and it has the therapeutic monoclonal antibody inside the nanoparticles. And first, this is a work that was done by Mor Sela. She's a former PhD student in our group, and now she's a postdoctoral fellow in Rice University. She's a really spectacular chemist, and she actually found a specific chemical formulation that allows you not to harm the proteins that are loaded in, and also to conjugate and have a protein inside and a protein outside, which is the targeting system out there. And what she found at the end of the day is having a particle with approximately 33 monoclonal antibodies inside, and on the outer surface, about 100 transferrin targeting moieties gave her the best results that she expected looking at the targeting. In order to prove that she actually has the targeting moiety on the outer surface, she again used nanotechnology. So on the surface of the transferrin, she conjugated gold nanoparticles, which are the black dots that you can actually see. These are liposomes, the antibodies inside, and the black dots that you can see here, those are gold nanoparticles that are conjugated to the transferrin, just to prove that the transferrin is really on the outer surface of these particles. But then when she went to the tissue culture, she didn't see any advantage in this targeting system. And we couldn't understand why. But then we understood actually that when we coat our nanoparticles, we use another molecule. It's called polyethylene glycol. There's a lot of discussion about this molecule today. And the polyethylene glycol on the outer surface of the liposome was inducing steric hindrance to our targeting moiety. So what Mor actually did is she used now a very short peg in order to stabilize the particle, but a very long peg in order to conjugate the transferrin. So now she has a stepwise system where she has the targeting moiety actually far out of the surface of the nanoparticles, and that actually allowed very good targeting into the brain. And since then, actually, we've been able, we use that, and many other groups actually use this approach in order to improve the targeting of the brain. So first we wanted to see inside neurons, once we enter a neuron, is our payload actually being released from the nanoparticles? And here, using advanced microscopy techniques, we actually were able to see that we have target engagement, meaning that the antibody adheres to our target, which is the alpha-synuclein inside the cell. Each one of the dots you can see on the bottom is actually a site in which the monoclonal antibody binds to its target inside the neuron. And that shows that we actually have not only uptake, but we also have release of the cargo, which is one of the confining things you need to have when you have a nanoparticle. Then we wanted to ask, using a BBB system, this is a supported BBB system together with collaborators, can we actually cross a BBB just in culture? And again, we saw very nicely that when we put our particles over a BBB, and then we run cryo-TEM of the liquid that's on the other side of the BBB, we find our nanoparticles that they crossed very well, and they even have the payload inside. Some of the particles stay on the BBB. And I always think about it like crossing a fence that has barbed wire. Some of your shirt may get caught on it, and those are the particles that stay there, but most of the people can actually just jump over and get in. So at this stage, what we wanted to do is show that we have therapeutic activity. And this work was done by Maria Polai. She just finished a postdoc in Harvard University and is working there in the biotech industry. And what Maria actually did is she injected a virus that over-encodes 4-alpha-synuclein inside the brain. And you can see here the right hemisphere of the brain, 2 weeks, 4 weeks, and 8 weeks after the injection of the virus. And this disease is a very, very robust model, and it actually damages also the dopaminergic neurons, which are a target for our therapeutics. If you look inside, this is almost scary. This is inside a neuron. You can actually see the alpha-synuclein aggregating throughout the neuron, and that and I think that's our target when we come to treat. So first we wanted to ask in a mouse, once we inject systemically the particles, do we reach the brain? And we saw high uptake into the brain of the particles after administration. We labeled them using a fluorescent dye, and we saw the particles also inside the brain tissue. But more than that, we wanted to see them extravasating from the blood vessels. And here, using with the collaboration of 2 groups, we were actually able to track the particles as they exit from the blood vessels inside the brain and enter into the brain parenchyma as we expected. And now we wanted to ask, do we get any therapeutic outcome? And that's actually our goal. And for that, we wanted to first see which cells we reach inside the brain. And of course, we see many of the particles in the endothelial layer, and that's the blood-brain barrier, the shirt that gets caught on the barbed wire. But we also see about 12% of the neurons taking up these particles, and that was exciting. Interestingly, the microglia and astrocytes had lower numbers of the nanoparticles. And later on, when we looked at this data, which of course was very surprising to us, we asked why, but then we understood that the transferrin receptor, which exists on the blood-brain barrier, actually also exists on the neurons. So we have a double advantage, one that we didn't know of a priori, but we saw it then from the results. So we wanted to go and see how can we affect the mice biochemically. And here, Patricia Mora worked on this. She actually took hundreds of slices of the brain, and she looked at the alpha-synuclein aggregation inside the brain. And what she saw after analyzing inside the cells and outside the cells is that mice that are injected with the brain-targeted liposomes, using it with a monoclonal antibody inside, we see a reduction in aggregation inside the brain. So actually the particles are crossing the blood-brain barrier, entering the neurons, and also reducing the aggregation, which for us was actually the target engagement we were looking for. But it wasn't really enough for us. We wanted to look also at activity of the mice. And Shai mentioned before also the rotarod. Here we ran several experiments, and one of them is the rotarod, which I'll show here. And we actually looked at the ability of mice that are either healthy mice, mice that have Parkinson's disease, mice that are treated with a free monoclonal antibody, or mice that are treated with the brain-targeted liposomes to stay on the rotarod. And of course, mice that have Parkinson's disease, of course, these also have Parkinson, fall off rather soon. They don't have the ability to stay on the rotarod, where the mice that actually are treated with brain-targeted liposomes, or the healthy ones, stay on for pretty much the same amount of time on the rotarod. And for us, this was actually, I think, a double way of looking at it. One, looking at the biochemistry, and two, also looking at the ability to penetrate into the brain. I promise this mouse also falls off. Statistically, they're all in the same range, and we ran this multiple times. I'll just give him the honor to jump off. Yeah, it'll happen in a sec. Okay. But then we met with the people for GRIN, and we were already thinking about, can you not only deliver a protein, can you also deliver a gene into the brain? And here we're looking actually at the delivery of messenger RNA into the brain. And this is work that was done by Gal Chen. She just finished our postdoc in her PhD in a group and starting a postdoc in Harvard. And Chaim was a PhD student in the group. And what they saw is that the particles that we were using actually for targeting the proteins need to be changed for delivering the messenger RNA. More specifically, the transferrin, which is a heavy protein on the particles, was less stable in the blood. And what they came with a very smart approach is they actually took small molecules that have receptors on the blood-brain barrier in order to cross into the brain. And they screened many, many different molecules. Some of them written here, such as acetylcholine, memantine, glucose, cocaine, and also molecules that were also mentioned here, throughout these couple of days. They conjugate them to the surface of the particles, and then they check the uptake into the brain. And they used a specific lipid formulation here that actually allowed also carrying messenger RNA into the brain, and then they test the encoding. So first they did this actually in cells. This paper actually just came out a couple weeks ago, and they saw that different targeting modes give us in the neurons actually different ability to express a protein. More specifically, they chose to go with acetylcholine, where they saw in many of their experiments an improvement in the ability of particles that have acetylcholine on their outer surface to penetrate into the brain and also to encode neurons. And what they saw is that when you take our nanoparticles and put them again on one side of a supported blood-brain barrier system, and then you look at the neurons that are underneath, you actually see high protein expression inside the neurons, meaning the particles are actually crossing from one side of the BBB to the other, entering the neurons and actually and then encoding them to produce a protein which was delivered through the nanoparticles. This next step was actually to go to the mice, and here again they ran a screen, and the acetylcholine actually won in this screen. And the interesting thing was, if you take just 3 of the molecules, just as an example, you take the acetylcholine, nicotine that was also on some of the particles or on target particles, you see very different distribution into the cells of the brain, meaning that you can actually direct the particles to different cell types inside the brain, where the acetylcholine gave us better uptake in neurons. Nicotine was taken up more by microglia, and untargeted particles we actually saw primarily in the endothelial layer of the cell of the brain, which makes a lot of sense. So we were asked here, can we actually show the engagement between the nanoparticle and the receptor? So here, together with a collaborator from Tel Aviv University, what we did is we encoded, actually they did the work of encoding, they encoded for an acetylcholine receptor on HEX cells, and it's important to mention here, Tommaso Patriarchi, he actually invented these receptors, which when you engage on them, you actually get fluorescent light. So you can prove the engagement. And what we saw is that when we put the acetylcholine particles on these cells, we actually see their engagement through the increase in fluorescence on the cells. And soon you'll see, this is when the particles are added, you actually see a very distinct increase in the fluorescence. And more than that, when you take the system and actually you look at the ability to encode for a protein inside, these are HEX now, not neurons, you start seeing actually that the particles engage with the acetylcholine receptor, and then you see the protein actually being expressed in the many different cells inside the culture. So just for us, it was nice to show that we actually have the ability to target using these systems. So we went on and we tested these actually in mice. We injected also locally and also systemically, which I showed you before. And the reason I'm showing the local administration is that we are getting more questions about local injection into the brain. I think for GRIN it's less relevant, but it is something that was on our mind. And there we see very nicely, actually, when we inject directly, we see expression in the neurons of our target system. So last night I was asked about AI, and I actually didn't intend to add these slides, but they said it's in the title, so I have to. And I want to say where we actually use AI in our system, and actually we're using it more and more in every line of our research. And this is actually a collaboration between us and Professor Kira Radinsky. She's an expert in artificial intelligence. She came to us a couple years ago and she asked, do we use AI in our system? And we said we don't. And then she taught me about this design, build, test, learn cycle, where you can actually inform the system with a lot of data and then actually start improving your experiments. And people were talking here a lot about the cost of experiments. And here you can actually also reduce not only the time, but also the amount of animals that you run in the experiments using artificial intelligence. And I'll just give one quick example. The team grew since then, myself, Roy, Yogev, and Kira. Maybe not the best images of all of us, but I just want to explain how we do it. So what we had is that we have in the lab only a certain amount of data, and we're also maybe in love with the systems that we have. But if we look actually at all the literature that was published on lipid nanoparticles for targeting different tissues, many people use many different systems. So what we did is we created bots that read all the papers. They read all the papers, all the patents, all the theses about lipid nanoparticles use. And these bots actually will correlate between a molecule. We write a word for the molecule, but then the particle will be a chapter, and the entire system may be a book. And we correlate that with activity. So we needed to write more bots because scientists usually present their data in graphs, but we needed to have that data actually as numbers. And also the biodistribution, which many of us show, usually we don't give the exact numbers, so we also had the bots read that. So now we have like thousands and thousands of data points, and that really improved our system. And we also created a lot of data internally. The reason we created data internally is that the papers, at least that we publish, is usually about our successes. And unfortunately, the failures, we don't talk about too much, the hard work. But we, in our real life, we learn a lot more from our failures than from our successes. But that doesn't exist. So we had to create a lot of failed data in order to teach the system. And I'll just give one example of how impressive it is. Here, for example, you can see delivery to the liver, to the lung, and to the spleen. The delivery to the lung is not very impressive. So we went back to the machine learning system, and we actually in vitro now, no experiment, no additional in vivo experiment. We started asking which problems are we maybe missing, which data are we missing here. We ran those experiments, we taught the system again, and we were actually able to change the data 100-fold. So this is before and this is after. Just to show two in vivo experiments, okay, one in July, one in October, and the huge difference, and I think that's something that we're learning that really can help a lot in our work in many, many ways. And we've done now this, we've tested the system also in mice and in non-human primates for targeting here, in this case, it's the lungs. Same work can be done also for the brain. I'd like to take my last couple of minutes just to talk about DNA delivery. Again, for us, it's very early, but it's GRIN2B, and again, what I hope I convinced you is that we can do delivery. And one thing that we can do actually, this is work that's been done by Mia and Shaked, they were here last year, and is actually they can load into their particles very, very large payloads, plasmids that are 60 kilobases, and they still see encoding. More than that, they can actually encode for multiple genes simultaneously inside the cell. This is just one example of encoding for different genes that actually reach different areas inside a cell. So these systems are really beautiful. The nanoparticles look slightly different with the DNA than our lipid nanoparticles with the mRNA. But what they did is they loaded the GRIN2B into a lipid nanoparticle, and then treated first. They treated here neurons, and they compared, in this case, mRNA. And this is the DNA on the left-hand side, and you can see that the mRNA seems more efficient inside the cells, but we do see encoding in both of them. But when we went on and we actually continued also this work, and then also inside primary neurons, in this case we're encoding now for GRIN2B, we see the encoding. And again, we're not experts in GRIN2B, and here we'll take any feedback and any suggestions you guys have, just to show that this is actually possible. I would like to end, this is the end of the conference, and to say that sometimes, we push, but we also need a pull. And the pull which we created inside the brain in order to pull our medicines in, one pull, we just published this, is music. And people mention music here, and I love really a lot of the holistic also approach that many of the people have. And what we did is we exposed also mice and also cells to different soundtracks. So if you could put on just soundtrack E, that's enough, because some of them are all a bit annoying. So soundtrack number E, yeah, thank you. So I assume many of you recognize Pink Floyd and The Wall, and the A, B, C, and D are, we just reported them, so you can listen to them. They're not as nice to listen, surely not when you're an MRI inside an MRI system, but we expose also cells, also mice, and also humans. In the cells and in the mice, we also exposed to nanoparticles. And now what we changed actually is that on the surface of the nanoparticles, we have sugar. So we're creating a draw because we're activating. When we listen to that music, unlike when you listen to me, where your brain is like, at least my brain would go like a bit tired. When you listen to music, our brain lights up, and that's what we actually saw. That when you expose the cells to the music, and also when you expose the mice to the music, we see higher uptake of the nanoparticles into the brain, or higher uptake of the medicines. And I totally believe that we can actually look at an era where we have combination systems, surely for treating maybe a GRIN patients, where we want to activate a specific area in the brain, we want the medicine to reach there, where it would actually sound a specific music, that area would light up because it's creating a demand, the particles are taken in, and then we actually, we have that encoding. So with that I will end. These are the people. I don't know if it's myself, I also wanted to do one of these, but just that you can see that under different musics, different areas, these are the ears, different areas of the brain are actually are activated. So what are the take-home messages for today? One, a big thank you for inviting me and for having me here give the last talk. I really appreciate that very much, especially coming from outside the field. For us, we do nanotechnology. We try to deliver medicines into the brain. We Uber drivers, not much more than that. And just give us passengers. That's what I want to say. We're looking for passengers, good ones, and those are the ones that we want to take. Thank you so much.